And let's see what it, okay. Let's see if it says,
Yep,
recording.
it says James is recording.
Yeah. Does it say recording on your end?
Mm-hmm.
Okay.
It does.
Thank you.
Okay.
Look at that. We figured it out.
Okay. Well, we may have to do this
Oh, wow.
again. We may have to do this again if we can't find the recording later.
That's right. That's right. Well, I think we'll probably be talking in the future about some mushrooms, maybe, anyways. So.
Okay, that'll be good. We can do it.
So, good morning. Thank you very much for joining us. I'm Terry Story, Stories in the Dirt, and this is Professor James White joining us. If you would tell us about you and what we're here to talk about today.
Yeah, hi. I'm a professor at Rutgers University, and I study, for about 40 years, I've been studying endophytic microbes of plants and how those microbes, how they go into the plant tissues, what those microbes are, and what benefits they give plants. And what I'm going to talk about today are endophytic microbes and a specific phenomenon called the rhizophagy cycle, where those microbes go into plants and carry nutrients into plants and give plants some other benefits. Okay, so that's what I'm going to talk about, and I'll go ahead and start if that's okay, Terry. I'll go ahead and start this slide share. Okay, so all right, so share. I'm sharing it, and go to there, and there we go. Okay, so the specific title is the importance of endophytes and the rhizophagy cycle for plant growth and health. And these microbes that I'm going to discuss are endophytes. And endo, this term actually means endo, meaning inside, phyte, meaning plant. And basically, these are microbes that go into plant tissues. And they can be fungi, they can be bacteria, they can also be algae. But mostly we're talking about fungi and bacteria. And in fact, bacterial endophytes are extremely common, more common than actually fungal endophytes. This is a picture of a fungal endophyte in a grass, and you may be familiar with these endophytes of grasses, of forage grasses and turf grasses, but they are these fungi that go into the tissues, and here you can see them stained blue, and there's little wavy lines here, and the arrows are indicating those, and you can see the plant cells in the background. So these go into the plant tissues, endophytes do, and they cause no disease. There's no destructive activity by these microbes at all. In fact, it's beneficial. And all plants have endophytes. It's very difficult to remove endophytes from plants. They all have endophytes, but you don't know they're there unless you have a microscope and you have special stains to see them. Endophytes are everywhere, and in fact, there are communities of endophytes in plants, not just one endophyte, but multiple ones. And many of these are bacteria, and these are, as I said, more common than fungal endophytes. This is an example. This is a plant that this is a Cadushy cactus on the desert island of Bonaire in the Dutch Antilles. But you can see these cacti are huge. This is a Cadushy cactus. And these Cadushy cacti carry endophytes as well. And if you look at their fruits, they produce seeds inside those fruits. And if you take those seeds out and then place them under clean conditions or aseptic conditions on something to germinate them, here you can see the seedling actually from one of those seeds. This one is stained so that we can see the bacteria inside, to see what these roots below, okay, those, and you can see the hairs coming out. Those are the root hairs. These roots and root hairs contain bacteria already. You won't see them, but we use special stains. And these stains, I kind of list them below. One is DAB for hydrogen peroxide, and NBT. We don't talk about ammonium molybdate for ethylene because these microbes produce ethylene, and I'll talk about that. Aniline blue for protein because these microbes will have protein in them, and you can stain them with these different kinds of stains. This is actually the root hair of one of those cacti. And the hairs, multiple hairs, you can see them crossing this screen here. And you see all of these structures inside. These are all the bacteria inside these hairs. These hairs are full of bacteria. These root hairs, these root cells, contain the bacteria. So they actually go inside of the root cells. This is a cell, a close-up of one of those root hairs. You can see the little kind of blue structures or dark structures, purple structures in there. Those are the bacteria inside that root hair. And you can see they're in pairs because they're dividing. Okay, what about crop plants? All crops will harbor endophytes. For example, here's cannabis, and I was a graduate student trying to work on hemp. And you can see these are the seeds here. If you germinate those seeds, a lot of these microbes are vectored in the seed. The plants will put the best microbes that benefit them on their seed so that when that seed germinates, the seedling will have those microbes to help it. If you germinate these seeds, this is actually the root hairs from one of those seedlings. And you can see over to the left, you see this hair, couple of hairs there, but you see all the brown structures in there. Those are all the bacteria. It's stained with that DAB for hydrogen peroxide. And that's staining there because the plant secretes hydrogen peroxide around the microbe or reactive oxygen around the microbe. And so you can pick it up. To the right, you see a hair, and you see all those little spherical structures in there. Those are the bacteria inside that hemp seedling root hair. Tomato, here's another example, tomato plant. You think tomatoes inside are going to be sterile. Well, in fact, they're not. They carry those endophytes inside. The fruits like this will have endophytes inside them. And take a look at those seeds actually inside the tomato. They will have microbes around them and actually already in them. And this is one of the microbes that we got. This is one called Micrococcus, but it produces these tetrads, these clusters of 4 cells. This is stained blue. They have little clusters there. And we like this microbe because we could track it because it has these clusters and it's very distinctive. So we use it experimentally and track it in the plant wherever we see these clusters. There's no other structure in a root cell that has that. So this is the root tip of a tomato seedling. And you see this area, you can see the hairs to the left over here, and you see the root tip, the meristem, actually root tip meristem over here to the right. This microbial entry zone is right here behind that meristem. And this orange purple area over here, that's where the microbes go into the cells. And this happens to be stained for ethylene, but we can see where those microbes are going in because they're producing ethylene. This actually, these images here show from an experiment where we put that Micrococcus, that tetrad forming bacterium, into Rumex crispus, which is a common weed that we have around. It's on farms and everywhere on the roadsides, but we use it experimentally because we can play with it and clean it of microbes and put microbes on it. And so what we did here is we put our Micrococcus on it, and then this, to the picture to the left, shows right around that microbial entry zone behind the meristem. And you can see the arrows are indicating those little tetrads, the bacteria inside the cells. Okay, and to the right, there is a plant or a seedling of that Rumex where we didn't put the bacteria in. And you can see, you don't see the bacteria in there, in those cells. That's in the same area, but without the bacteria. Okay, this is a close-up of that, just so you can see that. You can see the tetrads inside the root cells. So this is to show you that those bacteria go into those cells, and initially they will have their cell walls and they'll look like tetrads, but or whatever they are, rods, whatever they are. But eventually they'll lose that cell wall, that tetrad shape. They'll lose their cell walls and they'll form these protoplasts. And I'll show you that in a few slides. Where do these endophytes come from? They come from the soil. plants are fishing these bacteria out of the soil. They will take them out of the soil and they'll use them for acquiring nutrients. And the best ones they'll put on they'll move up to the top of the plant, to the flowers of the plant, and they'll put on their seeds so that the seedlings then will have those microbes. So because plants fish these microbes out of the soil, that's the reason we want to have good, healthy soils with lots of good diversity of microbes, the right kind of microbes for plants. Well, this internalization of microbes into plant roots was noticed back in 2010 by some Australian investigators. And this is one of those investigators, Channy Pong Fu, over to the right. They noticed that these bacteria and yeasts also were going into root cells. And they labeled those bacteria and yeasts with green fluorescent protein so that they didn't fluoresce when they looked at them under the microscope. And they can see the microbe they put in there and fed to the plant went into the root. And so this is some of the images from their paper. And if you look, for example, over here at D on this, you can see these cells with the yellow dots in there. That's bacteria inside the root cells there. And if you look at E, you can see a root hair there. And those are actually the little yellow or green fluorescent things are the yeasts going in to the root hairs or in the root hairs. And they noticed after 3 or 4 days that the fluorescence stopped and that the microbes seemed to disappear. They seemed to be degraded. And then they named this process rhizophagy, meaning rhizo for root, phagy for eating. And that is to denote the idea that these plants are eating these microbes with their roots, degrading them to get nutrients out of those microbes. So that's the rhizophagy concept. And we've been studying it since those investigators found that, maybe a little bit before. And we have concluded that it actually is a cyclic process. And that is that what plants are doing is they are attracting microbes to the roots with exudates. Their plants are secreting at their roots, they're secreting exudates right behind that meristem, that root tip meristem. And then that's attracting these microbes to the roots. And then they're internalizing those microbes into the roots. And I should say those exudates are made of sugars, they're made of organic acids, and they're made of amino acids. Okay, so they're nutrients that these microbes like. And so the microbes can go towards it, they move towards it, and then they're cultivated right there at the tip, and then they're internalized into the root cell. Once they go into the root cell, the plant secretes superoxide onto them. That's reactive oxygen. Superoxide is highly potent. It strips the cell walls off the microbes and it leaches nutrients out of the microbes. And some of those microbes break down, but some of them survive. And the surviving microbes in those root cells will trigger root hair elongation. And without those microbes inside those root cells, you have no root hairs forming, no root hairs will elongate. Okay, so with the microbes there, root hairs will elongate. And then as those root hairs elongate, those microbes are ejected back out into the soil. And then they reform their cell walls, and they go out and acquire more nutrients. And then they can be attracted back. Okay, so this we term the rhizophagy cycle. So it's the cycle between the soil where the microbes get nutrients and inside the plant, inside the root, where those nutrients are harvested. The rhizophagy cycle. So the rhizophagy cycle happens at root tips. So plants that have a lot of root tips will be doing rhizophagy cycle a lot. Plants that don't have many root tips will do it early in development. Later, the plants may develop mycorrhizae or something to acquire nutrients for them. But early on, all plants will use this rhizophagy cycle. And plants that have a lot of root tips do a lot of rhizophagy cycle. Plants, grasses, for example, forage plants, corn, other wheat, other kinds of grass plants will do this a lot. So this shows a root tip of an invasive plant. This is giant reed grass, or rather just common reed grass. This is Phragmites australis, one of the invasive plants. But this is a root, and you can see the root tip here, and you see this blue cloud outside it. That blue cloud are all the microbes. Those microbes are being cultivated in those root exudates by that plant. And then the plant will internalize some of those microbes into those root cells at the interface. And we don't exactly know the mechanism, how they're internalized. We don't understand that yet. There's a few hypotheses, but we won't go into that. But once those bacteria go into the root cells, the plant detects them and it hits them with superoxide. And they may go in, they may have their rod shape because of their cell wall, but after the superoxide hits them, it oxidizes off the cell walls, and then they form these protoplast phases over here to the right. You can see these protoplasts, and these protoplasts have no cell wall and they replicate very rapidly. And that's important because the plant actually will replicate these microbes inside the root cells. Okay, so the enzyme that produces the superoxide is this enzyme that's in the root cell membrane, root cell membrane, and that is this enzyme called NOx, or NADPH oxidase. And this enzyme actually will take oxygen from the air, produce superoxide, and that superoxide is what will hit those bacteria with. And I'll explain that more. This is from that common reed grass. This is a root cell. And you can see these arrows indicating the bacteria. These are already protoplasts. Okay, this was a rod, but now you see the protoplast phases. And you can see they're replicating in there. They're forming chains. The one to the right, you can see there's a big cell and a smaller one. And the very smallest is staining blue. It's staining blue because there's protein in it. The big ones are not staining blue because the proteins are being degraded. So many of these are breaking down as they're forming, and the oldest ones break down. So the plant can get nutrients out of these microbes, but it's still replicating. It's cloning these good microbes, and the location of these microbes is technically they're not in the cytoplasm. And this is just a diagram of a root cell. You can see the gray out here. That's the cell wall. And the white is the cytoplasm. So you can see it's not actually in the cytoplasm. Instead, they're in that space between the cell wall and the cytoplasm itself. And that space is called the periplasmic space. But it's really technically not in the cell, but it is underneath the cell wall. And those microbes can't get out. And the other thing that's going on is these plants are moving these microbes around. It's not just still. You think of cells as just sitting there, but in fact, they move. They're in a process called cyclosis. Those cells move the cytoplasm around and around and around in a circle. And it moves those microbes around. They're moving around. And I'll show you a picture of that later on. They're moving around in there, and it's at the same time hitting them with superoxide to extract nutrients from them. Okay, that movement is called cyclosis. Okay, this is just a picture. This is from a grass. This is a bluegrass. And you can see the root hair. You can see all these little spherical structures. Those, in fact, are the bacteria inside. This is a pseudomonad inside that hair. Here you can see, again, you see the bacteria inside the hair, the root hair of the bluegrass, but you can see they're beginning to accumulate right there at the tip too. This is what happens in the hairs. They accumulate at the tip, and that actually will trigger the elongation. I'll explain. That's a chemical process. We'll talk about that. But two things happen with these microbes are there. You get gravitropic response, that is, plant roots grow down with gravity into the soil. And also you get root hairs forming if the microbes are there in the cells. Root hairs form on the roots. But if you remove the microbes, you have no gravitropic response. Roots will stay on the surface of the soil, or they'll stay on the surface of the substrate, whatever it is. They go into the air sometimes, and the root hairs won't form. You don't have root hair formation. So those microbes are necessary for root hair formation and for the proper gravitropic response in those roots. This shows actually a Bermuda grass root in which we've removed all the microbes, and you can see the root here, and you can see the root older here to the right. You see there's no hairs on it. But when you put those microbes, when we put the microbes back on this Bermuda grass, you can see now, you can see the hairs form right away in there. So those microbes are necessary for hair formation. This is around where the microbes go in, and the white arrows are showing actually the lower white area here. You can see these rods in there, these brown rods. Those brown rods are the bacteria with their cell wall still on them. They just went in there. This is where they go in, and they still have the cell wall here. Later on, those cell walls will be oxidized off. This is a bit older, and you can see the hairs out here. You can see all the brown in there, all that brown. Those are microbes inside all those hairs, all those dots. These root cells become engorged by these microbes, by these bacteria. This is, again, that Pseudomonas that we're experimenting with. This shows a root hair, shows those microbes. They're in the hair. They're fat with microbes, loaded, all those little spherical structures. Those are all the microbes. This is from a tomato experiment where we removed the microbes using streptomycin antibiotic to kill the microbes. And you can see to the right, there's treated, and you see no hair. You see a root coming out, no hairs on it. And you see the same treatment where we did not use the antibiotic, and you can see the hairs at the beginning to form on that root of tomato. And this actually shows close up. Okay, if you look the picture to the right up here, that's what's called a root hair initial. And we treated this one with antibiotic, and you can see there's no microbes in that initial. So these initials form, but there's no microbes forming in there. And that initial, those hairs don't elongate. But to the left, you see over at A, over here, you can see that's a root hair initial, and you can see the bacteria in it, those microbes inside it. And you can see B beginning to elongate. You can see those microbes at the tip. And you can see at C, it's a bit longer, and you can see there's more microbes at the tip. So those microbes are actually being replicated. Okay, so one important question for us is, why is there this linkage between presence of microbes and elongation of the hair? So we're kind of fixated on this idea. And my student who's working on this is Ivy Chang, and we've been developing this for a number of months now, trying to figure this out. But this actually is a grass. This is Poa annua, a bluegrass, an annual bluegrass. It's a weed for most of us, but it's experimental for us. The root to the left shows a root of Poa annua with no microbes, and you see there's no hairs forming. And then when those microbes were put onto that seedling, you can see now hairs right away all over that root. And what we've been able to work out is our main hypothesis here is that these microbes are secreting ethylene, and we call it the ethylene hypothesis. They're secreting ethylene, and ethylene is also a plant hormone. Plants produce it. It's a growth hormone for plants. So these microbes are producing it, and it's causing growth in these root cells. And it's really, really fascinating. And we've developed this chemical equation to explain that. And I just want to go into this just briefly because it's exciting to us. But what's happening is these microbes are using an enzyme inside the microbe called microbial ethylene synthase. So they are, and that microbe, that enzyme in the microbe takes arginine, and it turns out that plants are providing the arginine in their exudates. Then they're coming from the root cells, they're secreting arginine. It's one of the enzymes that they're secreting. And so they can regulate this whole process. Plants are regulating this. Plants are in control of this process. So the microbe takes this arginine provided by the plant, and then it produces ethylene. It shows here the ethylene here in this equation, produces ethylene. The ethylene goes into the root cell, and it will trigger root cell growth. And it will also trigger superoxide secretion. We know that it triggers these two things because we can take an inhibitor, an arginine analog called L-name, and we can put it in the system, And it gunks up this microbial ethylene synthase, so it doesn't produce ethylene, and we also don't get superoxide being secreted. Okay, and so what happens is the plant normally will secrete, once it receives the ethylene trigger, it secretes superoxide. The superoxide will combine with the ethylene, will also strip cell walls off the microbes and keep extracting nutrients from them. But the combination of those two will produce hydrogen peroxide over here. And that hydrogen peroxide is what our stain, our DAB stain, that brown stain, that's what we're seeing over there. So that's okay. And this just depicts that. A little diagram shows our root cell again. And you can see the bacteria out here, the red, and you can see the ethylene secretion in there. So that ethylene secretion not only makes root hairs elongate, but it also makes root cells grow bigger. If we remove all the microbes, root cells are smaller and roots are smaller. And I'll show you that too. But this actually shows that tomato seedling again, and it is stained for ethylene, and it's kind of a purple blue stain that you get. And there are two places these microbes are going in. One is at the root cap, and you can see the purple out there at the root cap. And you can see this behind the meristem itself, this big area, this microbial entry zone. Well, that's also the cell elongation zone. Cells are elongating right in that zone. Okay, so this is from the root cap, and if you look at the root cap cells where microbes are going in, you can see these little spherical structures here inside the root cap cell, and you can see the purple around them. That purple is where the ethylene, stain for ethylene. You see the microbes and the ethylene in there. Okay, and this shows now where the microbes are entering behind the meristem, and you can see these little things out there. Those are microbes, little dots out there. Big arrows are indicating them. Going into the root cells here at that area. And if we look close at that, okay, this is a bit closer up. You can see the microbes actually already in the cell, and you can see the arrows there indicating those. And if we can go actually closer to that, and now you see here these cells here, you see the arrows. Now you see the bacteria inside the cell or in a little depression on the cell membrane, and you can see the purple, all that purple. That purple is the ethylene, that hormone, that microbe. And just imagine also, the plant is also moving these microbes around, so it's distributing that ethylene, making the cell grow evenly. So it's not just growing in one spot. This is an experiment using rice. And you can see this is with bacteria to the left here, E+. This is rice where we removed all the microbes and we put microbe on it, put back on it, this pseudomonad that we'd like to play with. And you can see how big these rice plants are. And we look to the right, we left the microbes off. There's repression there. They're not growing very well. And if you look, okay, if we look in this experiment in a clean culture, a xenic condition, not in soil where we can see the root, you can see no bacteria here. Over to the left, you can see this rice. You can see these roots are short. You can also see there's no root hairs on those roots. And what we did here, instead of putting microbes on now, to test our ethylene hypothesis, we put these seedlings in ethylene gas. We put them in a chamber and we put 1 part per million ethylene gas. And now you can see that's over to the right. And now you can see the roots are longer in that ethylene gas, and you can see they have hairs on them. So we can replace those microbes with ethylene gas and recover development of those roots. Well, those microbes, the formation of root hairs are important because that's how those microbes are ejected back out of the soil from the root hairs, from the tips of the root hairs. This actually shows a root hair. And here you can see actually the microbes, this cluster of microbes, the big arrow where they're ejected. And you can see inside the root hair there, you can see there's actually microbes there in that tip. This shows another one. This is a sedge, and you can see the little dots in there. These hairs get fat with these microbes. In fact, the reason these hairs are fat is because they have microbes in them. These hairs, and naturally without microbes, the little root hairs are skinny, little skinny things. But because of the microbes there, and you see the ones that are stained, but even where it's not stained, those are microbes in there. And you can see outside to the left, where the arrow is indicating, those are all the microbes that came out and they're reforming their cell wall after they're ejected out there. And what we think is going on is that this ejection, this microbial ejection, is happening when these hairs are elongating. And what we think is happening is that the microbes in mass are secreting ethylene. That's causing the hairs to elongate. And when the hairs elongate, it gives a pulse, a growth spurt, and then that shoots some of the microbes out. And then that takes a while for those microbes to be built up again, and they're cloned in there, and then they're built up, and then it causes a spurt again, and you keep going. It keeps going like this. And you see this. This is actually a root hair that shows where some of the microbes remain on the inside of the hair after each spurt. But you can see it's like periodic spurting ejection of those microbes, leaving those microbes the kind of almost like remnants of those microbes inside those hairs. Okay, so we think they get ejected every time there's a growth spurt, and that those microbes themselves are causing that growth spurt to happen. So, okay, this is actually one of the sedges, and this is from Bonaire. It's growing in the limestone rocks. This sedge actually does not have any soil or very much soil, but it does have a lot of microbes. And this is actually a picture below that shows that sedge root hair. And you can see the microbes staining kind of red-brown around the periphery of this hairs. Okay, and now to the upper image, what you see is this is a movie from a living root hair. And what you see is the cyclosis that's happening that I talked about, this constant movement. These hairs are in motion. They're not still. They're in constant motion. And so these microbes are being moved around and constantly being hit with superoxide. That's important, we think, because for rhizophagy cycle, for nutrient acquisition, what this does is it will break down gradients that happen, nutrient gradients that happen between the microbe and the plant. If those gradients happen, there's slow nutrient transfer between the microbe and the plant. But because it breaks them down, nutrients are exchanged very rapidly. The other thing this does is it will continually move the microbes around and keep them so that they can't burrow their way out, so that they can't get out of the hairs. They can't get out of the sides, for example, produce enzymes and escape. And they're also broken up and replicates. And this is one of the things that helps replicate them by breaking them into little pieces as it keeps moving them around. And here you can see actually a hair, and you see all of these probably originate from a single cell, a single bacterium cell. And then all these other microbes around it, all these other spherical things are progeny of that. So the plant is replicating these microbes. Again, the plant is in control of this whole process of these microbes. This is how plants naturally cultivate these microbes and get their nutrients without inorganic fertilizers. This actually shows a root hair. And you can this is a sedge root hair. You can see that cyclosis again, that movement, and you can see the microbes jostling around at the tip, accumulating at the tip. And again, as that grows, they're ejected out of that hair. And this actually shows some of that ejection. And this is an image. This is tomato. And you can see what happens is there's a swelling at the base of that hair, and it will go to the tip. And as that swelling goes, and this is how hairs grow too, is they get this swelling and they push forward like that, and they get a little bit longer each time. But you can see these microbes are ejected, and they're ejected out of pores at the tip. You can see if you look at 3 over there, you see 3 little pores right at the tip. And when these hairs are growing, they naturally will, because they're growing right at the tip, they naturally will break and have pores forming. And so those protoplasts will be squeezed right out of there. Okay, this is just a diagram that shows that. We call that the cyclosis expansion wave mechanism for microbe expulsion from root hairs. But it involves the cyclosis, moving those microbes around, accumulating at the tip. And then there's an expansion wave, a growth spurt that happens. These microbes are secreting ethylene, and that causes this growth spurt. And then you have this expansion that happens and squeezes those microbes out. Okay, and what happens then after the microbes are squeezed out? This shows to the image to the right. These are, in fact, this image shows root hairs. And you can see the protoplasts. To the left, you can see the little spherical things. Those are the protoplasts inside. And to the right, actually, you can see some protoplasts inside, but mostly you see, this is a yeast, actually. You can see these yeasts coming out. You can see the smallest ones are at the surface, right at the tip, and the oldest ones later. That's because they will actually grow right after they're ejected out. What happens is after that ethylene pulse and growth expansion, exudates come right out of these root hair tips and feed these microbes. And so those microbes are made competent for soil living again, and they can go back out into the soil and acquire more nutrients. This happens to be a yeast. This is a Rhodotorula for anyone who's into yeasts. So root hairs, what is the function of root hairs? We've always heard that root hairs absorb nutrients, and that's probably true. But in terms of the rhizophagy cycle, root hairs function to move those microbes out into the rhizosphere. It wouldn't do any good to dump them right on the surface of the root, but to move them into the rhizosphere and put them out into the rhizosphere so that they can go and acquire more nutrients. And this actually, to the image to the middle here, just shows some of the hairs. You can see those, this is a fluorescent stain. You can see those microbes coming out of those hairs. And these absorb nutrients. That's the function of the rhizophagy cycle. And I'm just very fast, I'll go over that. And this is some work done by one of my visiting scientists in the lab, Shuai Zhang. And what he did is he worked on wheat. He removed all the microbes off the wheat, sterilized them, and then he put them on one at a time, and then measured nutrients that went into the once he grew the wheat with the microbes, then measured the nutrients that come out of the soil. Okay, and this actually this shows some of the plants without bacteria. You can see these have little stunted roots, and also the shoots are quite small. And to the right, this is when he put one bacterium back on. These are the same age and the same experiment. You see the roots are much bigger, like 3, 4, 5 times bigger. And you can see the shoots are bigger too, but you really see huge differences in roots growth. Okay, then he measured nutrients inside the shoots. And this is for nitrogen. You see no bacteria to the left. You see when he put the bacteria on, 3 different bacteria, and then measured nitrogen in the shoot, you can see, about 30% more nitrogen. So it's not like microbes deliver all the nutrients that plants need. I mean, some of these nutrients can come from soil absorption without microbes. Okay, and that's why we can put nitrogen fertilizer on. But plants doing this rhizophagy cycle can get about 30% more nitrogen than plants who aren't doing it. This is the natural way that plants work. This is the way that they get their nutrients, is through these microbes. And phosphorus, it looks like about 20% more. You can see to the left with no bacteria, and then to the right, you see potassium. You can see this first microbe happens to be a bacillus species. Looks like it's like 3 times the potassium. Certain microbes deliver a lot of potassium to plants. And that's important because, we think that's important because potassium is one of the nutrients that you would expect to come out of these microbes right out of their cytoplasm when they're being oxidized. They leak potassium. That's one of the first nutrients. But you see different microbes give you different amounts of potassium. And that's an important phenomenon. Calcium. Okay, you can see microbes deliver sulfur. Microbes deliver more than without. Manganese. Microbes are delivering more. Magnesium, zinc, and so forth. And strains are unique. Not all strains of microbes are equal in terms of the nutrients that they deliver. Okay, so it looks like, okay, so I want to summarize by indicating that it's not just nutrients that plants get by doing this rhizophagy process. Because the microbes go into the plant and they interact with plants using reactive oxygen, plants upregulate oxidative stress genes. So then plants become hardier. They become resistant to drought, which is an oxidative stress. They become resistant to heavy metals. They become resistant to heat. Pathogens also. some of these rhizophagy microbes, bacteria, will go out into the soil and they'll colonize these pathogenic fungi, and they'll drain those pathogenic fungi of nutrients. They'll make their hyphae leak nutrients, and those fungi will become weakened, and they won't cause disease. They'll become avirulent. So they actually benefit plants in protecting them from disease. These are some papers that you could go to. These are online papers. One of them published in Microorganisms on the rhizophagy cycle. You could Google that. Another also, they're both open access. Another one is a review article, Endophytic Microbes and Their Potential Applications in Crop Management. It's in the Journal of Pest Management Science. And you can find that also online if you want to. So I think that is it. And Terry, I think I'm ready for any discussion that we can do. I hope it wasn't too long. I don't know how long it was.
I honestly don't know either. I enjoy it. I enjoy your conversations. So maybe other people can't sit that long. I enjoy listening to long conversations. So my questions are around how these things, this information can be applicable or beneficial to someone who's raising a garden or a pasture. So I want to go to your conversation about, or your comments about how the plants takes those bacteria and they bring it to the seed, right?
Yeah.
So as in a pasture, if I'm understanding this correctly, those nutrients are starting off with the plant, right?
Yeah.
My question is, if I have more species of plants that are starting off with the healthy seed, are they going to benefit each other by sharing those microbes, or does it make a big difference?
We think it does. We think that, some microbes are not shared between all plants, but they're more specific to a species. But there are many microbes that can go between plants. And certainly when you have a mixed plant community, you increase your microbial diversity, and your plants are going to benefit from that increased microbial diversity. They'll benefit in some cases. They may benefit by sharing nutrients. Some plants can, the microbes from one plant can go over to your pasture grass, and your pasture grass can absorb those microbes and get those nutrients from them. And they may have, those nutrients may have originated around your other plants that you have growing in the pasture. So, and in general, your community diversity of microbes in the soil is going to be higher when you have a mixed, diverse plant community growing on that soil. So yeah, we think that diversity may be a benefit to your pasture plants. Now, there is likely a literature about this already that you can go into, where people have studied mixed community of plants versus a single stand of one grass, and you can get some more specifics about what's going on. But I mean, we think it does have to do with microbial diversity in the soil. Now, the question also of seed transmission, that was a good question. Seed transmission. These microbes, some of these microbes do go to the seed. And so when you put your seed down, your seedlings will do better when those microbial populations are fully developed on the seed. In fact, we did a study where grass seed that is produced in moist environments will have a more diverse microbial community that goes onto those seeds and that develops, because you have, what happens is a diverse, is this microbial community on seeds as they mature, takes a certain amount of moisture and takes some time to develop. And if you take those seeds and put them in a very, very dry environment, when they develop, you don't have enough moisture for the microbial community to develop. And when that happens, when you don't have that microbial community there, the stands tend to be not as even, because the microbes, the biostimulant microbes, the protective microbes aren't present. And so you have more damping off and you have other problems in your seed. And in some cases, people treat their seeds. For example, with Bermuda grass, they will remove, the microbes are actually carried on the hulls, or that cover the seed itself. And so people then will remove those hulls and they'll put chemicals on the surface. I think it's clay on, and then they'll embed that with either fertilizers and sometimes fungicides to try to control disease. But if you have the microbes there, those seedlings you won't need all that, the fungicides, because that's going to control disease, and you won't need the fertilizers as much either, because the microbes are there and they'll get their nutrients from those microbes. The other issue is for gardeners is you want to actually, the main take-home lesson is you want a healthy soil. You want a soil that has a good amount of organic nutrients in it that will support these microbes, and that the plants can then use the microbes to acquire nutrients from that organic material. So did that answer the question, Carrie?
It did. So on that note, I'm considering experimenting with some ferments, making my own inoculants, I guess, experimenting with that this year. And my question is if I'm on the right path with my thinking. So if I take like the fruit of a tomato, right, remove my seeds and put them into storage, and then take the remnants of the tomato and ferment that either in a water or something else, store it, would that be active later? And could I use it to inoculate those seeds in the beginning of the year to help kind of bring that along?
So, I mean, that's a good question. It will change. You do a ferment and then you put that away. The ferment, you're going to have a succession of species that happen. And this is, well, this is a perpetual problem, really, with people trying to apply microbes and liquid over you need. This is why most of the companies use bacillus or endospore forming microbes, because they'll form endospores and they can stabilize, right? But with a lot of these microbes, undefined community of microbes, you're going to have a constant change over time of what's in that. That doesn't mean that what's in it won't be helpful. It may be helpful. Now, if you do a ferment and you put it under refrigeration, maybe, and lower the temperature to a very low level so that growth, metabolic activity is highly reduced, you might be able to do exactly what you said and have, what you should do. You should, I saw your online talk where you talked about experimentation, right? You should experiment with this. You should do an experiment. You should do an experiment. You should go in and you should do 3 things. You should do where you don't apply your ferment, where you apply your ferment that you, maybe that's fresh, that you apply ferment that you stored for 2 months or a month in refrigeration, and that you just let the ferment just change and continue to ferment, over a period of time, over a longer period of time, and then apply all 3 and see what you get, because there may be a difference, and you might find that 1 or more of those treatments is going to be beneficial. It's going to be beneficial. I should say, I should add something here. I mean, it is interesting, the ferment idea. You know, the American Indians, for growing corn, they had another way to do it. They actually they would take the microbes off of wild, this was what they called the corn medicine. I think it was the Iroquois that would do it, but they would go get wild grasses like Phragmites, the native Phragmites, and Hystrix, Elymus, some of the woodland grasses. Then they would take those roots and they would put them in water and they would beat them up and they would heat them up a little bit and get out bacillus and some other species that were resistant to their warming. And then they would take that and then they would germinate their corn, because even they lost microbes in corn because they would collect their seeds and store it. But they would take that corn, germinate it in that liquid with other microbes, you know, and they called it corn medicine. It was magic. But they were working with microbes. And then once it germinated a little bit, it was colonized by those microbes, and then they would go plant that in their corn mounds. And they noticed that their corn medicine made their corn grow better. And so they were doing something that we in modern agriculture aren't doing. We have another way. Instead, you know, I mean, theirs is probably better. You know, they get all these microbes from the roots of these wild plants, while we have, you know, we've selected, we maybe get 4 or 5 different microbes and put that on. So anyways, that's like your fermentations, really. You get these undefined communities and put those on. And yeah, so you have to experiment with that.
I plan to. It'll be
Yes.
fun.
Yes. Let me know how it goes. Let me know how that goes.
I will. Let's see. So those root hair developments, you put on your experiment antibiotics, right? To sterilize them, control.
Yes.
How do synthetic amendments affect that root hair development? And I don't really want to be specific because I've never used any kind of synthetic amendments in my gardens, so I don't want to misspeak.
Well, they meaning, like, for example, ammonium or other fertilizer or other chemicals, right? Okay, so if you're inhibiting the microbes, like weed control, for weed control, glyphosate or something like that, we know that glyphosate it has an antimicrobial effect, and it can impact your microbial populations. So you can impact the rhizophagy cycle if you're hitting that community. Now, I mean, that's undefined ways. You know, we don't exactly know what microbes are going to be inhibited. If you're putting, you know, the worst thing that you could do is to put soil sterilants down, you know, something metabromide or some other kind of sterilant that people use these days to kill everything. That would be the worst thing that you could do, because you just kill everything. Or if you do deep turning, that would also impact the microbial community. It would make it so that your roots don't have access to the healthy community. Instead, you buried it deep down. The other applications, like, for example, you're putting ammonium down, it's known that you inhibit the nitrogen fixation and nitrogenase in the soil. So these microbes aren't able to get their own nitrogen. So you reduce that component. And it's also known that you reduce exudation to some extent and the cultivation of those microbes. So you're not attracting the microbes as much. So, I mean, you are impacting the rhizophagy cycle, you know, I mean, to differing extents. Now, sometimes people have to do that because they have poor soils, they have only sand. And so they're, you know, until they build up their soils and the soil community and and make it a healthy soil with a healthy microbial community, then they've got to do something or they have no crops. But if at all possible, it's better to build up your soil and consider the microbial community when you do that. Consider this is what plants do. Healthy plants do this, and it makes them even healthier when they do this and hardier. Yeah. Was that a good enough answer?
It was excellent. Thank you.
You're welcome.
So as far as compaction goes, because we need oxygen, right?
Mm-hmm.
And we need pore space for everything to be moving around those roots, right?
Mm-hmm.
Organic matter is probably where I'm going with that, creating that pore space. I right here where I am have a very chert-based soil. It's very easily compacted, and we have had, my first year here, I walked the property every day, and I saw in every plant a nutrient deficiency. And I am equating that to the compacted soil, because the plants are not able to access, it's not working. The soil's just not working, period.
So I can address that.
Okay.
You saw when I showed you that where the superoxide comes from, and it's coming from this enzyme called NOx in the root cells, right? And that's what the plant is using, the superoxide, to extract nutrients from these microbes. Okay, so if there's not enough oxygen in the soil, say for example, the soil is very compact and it gets moist and you don't have the air spaces in there, there's not oxygen there. And also the CO2 levels may go very high because the CO2 can't get out of the soil. So under those conditions, the rhizophagy cycle gets inhibited. In fact, we can, if we use carbon dioxide, we can put carbon dioxide in very high levels, but we can put carbon dioxide around these roots, and we can inhibit the rhizophagy cycle. We can inhibit everything. When we do that, we can inhibit root hair formation, we inhibit extraction of nutrients, we can inhibit everything. So without good aeration in the soil, the rhizophagy cycle can be paralyzed. So, I mean, is that exactly what ended up why you got nutrient deficiencies? I can't say, but it would affect it.
Yeah. I've been running experiments on the property. So I've done deep litter in my chicken and my pig pens, and then spread that out onto the test field. I had one 25 by 100 foot test field that I put that on, and then the other half 25 by 100 foot I didn't do anything, just to see how it would compare. Mm-hmm. And then by the middle of the summer, the grass was so thick where I had put that, it was probably hay and grass cuttings and straw and manure. I put that over there, and then cut it all back at the end of the summer and put another layer of that litter over the top of it. It was all munched up and kind of mulched. Yeah. Okay. And then I tarped it over. I went and pulled it back last week to take a look at it, and it's full of organic material, and you could put your hand into it now. So my goal, I think, is to create that pore space, right, with the organic matter. Just, I don't want to misspeak because this is not my forte. I'm just a gardener trying to figure things out. But I like understanding some of those mechanisms that are happening under the ground for my plants in the future. So
two things. Okay, the aeration, we talked about that. You need that aeration. Those, but also the microbes, with getting that organic matter in there, you're making food for microbes. Microbes love that, and so you're improving the health of your soil. And the microbial, there is a way of a stream of thinking in agriculture, regenerative agriculture or biological agriculture, where almost everything goes back to the microbes. And so, I mean, if you think along those lines, you know, you want to have that, all of that organic material in the soil, mixed up in your soil, so that those microbes have the right food to develop and then carry nutrients to your plants. But we also know that you need oxygen in order for the rhizophagy cycle to work. So you're making that aeration, making that soil so you have aeration, air in it. So yeah, I mean, you're doing. I mean, I'm only a lab scientist too, right? So I'm not an expert on what you're an expert on. So, you know, I mean, I think that that's the right thing to do, what you're doing.
Yeah, this is my first time working with compacted soils. I've lived all over and mostly been a backyard gardener, which, you know, that's generally imported soil, or it's been topsoil that's been brought in after the fact. So this is my first time dealing with, you know, old agricultural land that was plowed and tilled over. I don't even know how many times for how many years. Probably 20 or 30 years ago, it was turned into a small farm lot and hasn't been plowed since. But it's really compacted. There's no topsoil to speak of when I first came in here to look at it, which is why I decided to start doing the composting and the cover crop and the, you know, just trying to build it all up and get the soil, get the roots digging down into it. Because if you dig down at all, you get compacted like almost immediately. If you anywhere that I haven't experimented yet, it's hard. When I get to the areas that I've experimented in, I'm getting down inches now before I'm starting to hit that hard pan. And so now this year, my goal is to start getting plants in there to break up, right? And just start reaching farther down into the ground to create more topsoil. So
It's kind of amazing, really, how, you know, one way of thinking and industrial agriculture has destroyed much of our soils. It's really amazing. And I mean, it's, and you know, the problem is, once you do that to soil, you really have to get radical to bring it back to life. And you essentially are addicted to chemical fertilizers, and because plants won't grow without it. Now that you've done that, you know, now that we have the hard pan, now that we have no organic material there, you're totally dependent on those kind of fertilizers. So it takes really, I'm going to say, healing the land, right? Healing the land. I mean, it sounds mystical, really, but that's essentially what you have to do to bring it back. You've got to heal that land, heal that soil.
it's very interesting. I've never been a mystical type person, but I do find in a lot of the different experiments and the different articles that I'm reading, it is kind of some of the things I remember hearing from my more hippier dippier associates or friends of family or whoever along my long and sordid past. Some of those things are ringing more true than I expected them to. You know, I was listening to a really interesting conversation about the using magnetics in the water, right? And I remember people when I was young talking about magnets and all these other mystical things. I never thought there was anything to that, but now I have to think about it because there may be something to that.
Well, I want to add to that. We don't know the mechanism that plants use to internalize these microbes. And there are, while I don't know a lot, I don't know much about it. There may be magnetic currents that happen inside roots and that enables them to internalize these microbes. And we know that this, reactive oxygen has charges with it. And, what we're seeing are a lot of charged particles. And those charges might, there's kind of electric, and I have read that there are some currents that happen on roots around the meristems, and that's exactly the area where these microbes are being taken in. So there could be something to, that has to do with magnetism in terms of, and I can't tell you, it's not like a crystal. It's not like a crystal, you're in crystal. I'll hold my crystal up. It sounds kind of crazy, but I mean, actually, there may be some electrical currents on these roots, and that may have to do with internalizing. We know we can take, for example, nanoparticles, silica nanoparticles, and put them around these roots, and they will internalize those nanoparticles, which, that's amazing. That they do that. How exactly is that happening? We still don't know that. Maybe they're just being pressed in. Maybe there's some electrostatic charge that pulls them in, just like microbes are pulled in. We don't yet understand that. That's another thing. We're not used to looking at these electrical currents and these little tiny little things like roots. We do know. I have done some experiments actually with larger roots. And with larger roots, you can, for example, one of them was, remember the potato experiment where you take a potato and you put little electrodes in it? Remember that? And you can get a charge across that potato. They almost like a magnet. That happens to not be a root. That potato happens to be a stem, but you see the same thing in roots. And we've done experiments with some larger roots, with mangrove roots, and we can measure the charge from inside the root to outside the root. And so that charge differential makes it like a battery, and you can develop a current that goes from outside of that root to inside that root. So this could actually be happening in these tiny little root tips. And so magnetism might have, those people, while they seem really flaky to me, there's some scientific basis for not totally disregarding some of those ideas, being a little bit open to it, not wholeheartedly embracing it, but yet not dismissing it out of pocket.
Right. Yeah. Well, I plan to have some experiments. I'm gonna fiddle around with all kinds of. I've stumbled into this with the pandemic. All kinds of information is just bubbling to the surface, and I will of course try to focus some of my experiments. But I'm enjoying the access to all these different ideas. I plan to take some of them and put them onto my property and see what we can get done. And I'm kind of rambling a little bit now, but to focus, it's really exciting to have so much information available. I have been trying to find some of this information out as a layperson, gardener, trying to figure out what's going on with my own garden. And in the last 2 years, a lot of information has come available and become really accessible. And in the last year, super accessible. It's just, how did I describe it? I'm like a pig in mud. I have been thoroughly engaged this last year in all the information I've had access to. So
I would add that it really, as we learn more about this, it's like a new world, and the world, in terms of agriculture, we all used to think, oh, it's all about, providing inorganic fertilizers and making plants grow, and that's all it is. But this is a whole new biological world that, it was some of the old ways, like what I talked about, the corn medicine of the Iroquois, right? I mean, and your fermentation experiments using ferments, it's rediscovering what the Indians used to do for their agriculture that we've forgotten in our modern approach to the world. Everything is so perfectly defined chemically. It's a chemical world, but it's more and more, it's really more biological, and also biological as well as chemical.
Right. And physical. I've had to
Yes.
dust off the old thinking cap from, way back when.
Yeah. Right.
Yeah.
Right.
So I asked about soil space. So you had mentioned hydrogen peroxide. I think probably this will be my last question. I have heard in some conversations people use hydrogen peroxide in their fields. I think I may have gleaned a reason why in your conversation. And can you maybe elaborate on that a little bit?
Well, that's it. Why they use it in their fields. I'm thinking they're using it because it will liberate proteins out of some bacteria. It will kill some of the bacteria, and you'll liberate those proteins, and the plants can absorb those, or some of those broken down proteins, plants can actually absorb them, or other microbes can get them and carry them to plants. Hydrogen peroxide will kill some organisms, some if they don't have catalases, right? But others just break it down, so it becomes broken down. But you'll also get you'll get more oxygen, for one thing. If hydrogen peroxide, when it breaks down, you can get some oxygen liberated from that in that process, and so that may aerate the soil a little bit. Yeah, so I mean, maybe the oxygen could favor rhizophagy cycle if you have extra oxygen in the soil. That could be one of the effects. But the other effects is you may be killing some microbes, and I don't know how much they're putting in. Yeah, so it probably doesn't last long when they put it on, though, because you have lots of yeasts and lots of fungi there that'll be breaking it down wholesale very rapidly. So you may not have much of an effect on your microbial community, but you might produce oxygen that then would favor a rhizophagy cycle.
So it'd be, if somebody chose to use it, they'd really want to know what their objective was.
I think I go back to what you said with your experimentation, and I'm personally, just like you, a strong proponent in experimenting, and I think that growers also are experimenters and should be experimenters. If they're taking the word of a company representative or a county agent, I think they should listen to those people, but they should test themselves. Don't take anybody's word, as the law, but experiment with it.
Would you have any advice for a gardener turned homesteader turned experimenter?
Well, my advice would be keep doing, keep doing, keep doing, keep being open, keep exploring, growing. I think we're on the edge of new understandings of how agriculture can be done, that will preserve the environment and make plants hardier and healthier for people. So I think keep growing. That's my advice. Keep growing, keep being open. Don't believe everything people tell you. but
Absolutely.
Healthy skepticism, is always okay. Don't cling to the old ideas, but don't jump off the cliff either. Yeah.
Right.
So that's my advice.
Well, I probably have taken up more than enough of your time, and thank you very much for visiting with me.
My pleasure.
We'll see you again here in a few weeks. And thank you for your time.
It's my pleasure, Terry. My pleasure. And so I think we want to stop sharing somehow, but we want to make sure we have
I think just stop recording.
I'm afraid I don't want to go end. I just go to recording. Okay, pause, stop, pause, stop. Okay, I'm going to push it.